Okay, well, thank you very much. I was delighted to receive an invitation to give this little talk. And I think some of you, I know a fair number of you, I hope you know me, but I'm going to reveal some things about myself that even the people who know me and have worked with me probably didn't know. Title, The Long and Winding Road, My Life with Myxomycetes. And my road has not been a straight road with respect to myxomycetes because I haven't always been a myxo person. Before you start booing and cheering, well, there are other reasons. You look at that and say, what does that have to do with a myxomycete? Well, actually, a lot long term, but I started out as a forest ecologist. When I went to college as an undergraduate, I don't know that I ever heard the term myxomycete. You could have hit me with one and I wouldn't have known what it was. I hope I know a little bit more now, but when I started out, that was not the case. I did my graduate work at a place called Virginia Tech, which is in southwestern Virginia. You can see Virginia on the map. If you look at that map, those are the Appalachians, the major mountain range, mountain system in Eastern North America. It actually has three components, three parts. The very eastern part is called the Blue Ridge. The central part is called the Ridge and Valley. And the most western part, when you get over to West Virginia, is called the Appalachian Plateau. This is the Ridge and Valley, and this is where, well, that's where I did field work to get my master's degree and ultimately my PhD. That's a ridge in the Ridge and Valley. You see it's longitudinal. You can see the one over the top, or you can see several of these. On the slopes of those ridges are predominantly oak, quercus forest. Not all quercus, but certainly a large component. That's me a little younger, in fact, a lot younger. And the lady standing beside me is my wife, who has been my field assistant since we got married many, many years ago. This is actually the Appalachian Plateau. It looks pretty rugged. It is pretty rugged. For those of you who have done field work in mountainous or montane areas, it's kind of tough getting up and down the slope. At the top of some of those slopes are coniferous forests, actually spruce, a little bit of fir, but mostly spruce forest. A very different forest type or ecosystem type, whichever term you want to use. On those slopes, especially in the Ridge and Valley, there was a lot of a special kind of tree, special to me at least, called American chestnut. Healthy, grew very tall, used for just about everything. But about 1900, a fungus was introduced into North America from Asia. Unfortunately, it killed American chestnut. It was called the chestnut blight or chestnut blight fungus. I got my master's degree working on post-blight infection or post-blight composition of formerly chestnut-dominated forests. So that subject is very dear to me. This is what happened after the blight had gone through. That shot was taken in about 1932 on one of those slopes. Those dead trees are all chestnut trees. Appalling. Well, when I started publishing, you look at that and you say, well, present distribute, it doesn't mention myxos at all. No, it does not. I was not publishing on myxos at that time. Well, quite frankly, when I did the field work, I wasn't. I did have a few minor publications after that. In graduate school, I was introduced to the fungi, the macrofungi, by Orson Miller. Again, that's my wife there. That's Orson's wife, Hope. Orson was the one, he was actually placed on my graduate committee, and he was the one that suggested that I take his mycology course. And I tell people, I didn't like the looks of that. I had no interest in taking a mycology course, but I took it and I fell in love. Not with my wife. I fell in love with macrofungi. I still love them. Maybe they're not as pretty as myxos, or maybe not. They're neat. I think they're neat, and some of you are likely to agree with me. Wow, that's a neat one. Well, I started writing, and I've done a lot of writing in my career. I like to write. I have no trouble writing. And you look at these and say, well, again, where are the myxomycetes? No, I've actually published fairly extensively on macrofungi. But you say, well, finally, a myxomycete. That's what he's supposed to be talking about. Well, yeah, I guess. A nice Arceria. Well, there are a number of things, number of events, number of things that occurred in my life that sort of bumps that, I mean, you're driving down the road and you hit a bump, it alters your path. Well, the same thing happened for me in terms of my career. I had a postdoc, a summer postdoc in 1982 at the University of Virginia Mountain Lake Biological Station to do research. What I did is I started what I consider a fairly major project looking at the ecology of myxomycetes. I worked on that for 5 years during the summers. There were 5 study areas, more than 1,700 field collections, 1,500 collections, moist chamber cultures. And as an ecologist, remember I came into myxomycetology, if you want to use that term, as an ecologist. I wanted to look at the factors, the environmental factors that were affecting the distribution of myxomycetes. Well, what can you do? I tried to determine all of these. Some of them didn't really work out, and some of them I think did, but the composition of the vegetation, which I found out to be a major factor, by the way. Light intensity, I measured that for the 5 different communities, but didn't get a lot out of it. Relative humidity, wood decay rates, wood moisture, pH, microclimate, in other words, the air temperature, litter dry mass, because I'm looking at the myxos, as indicated earlier, using moist chamber cultures and field collections. I even looked at soil chemistry. This paper came out of that, and at the time I was very proud of it. Distributed ecology, you can read the rest of it. I think at the time it was the most comprehensive study of myxomycete ecology ever published. I could be wrong, but I don't know of any others because I measured or tried to quantify so many factors. This was followed up by a second paper. The first paper essentially considered only the field collections. This one, moist chamber culture collections. And I tried a few things, again, coming from ecology. I tried ordination, detrended correspondence analysis, in other words, gradient analysis, looking at distribution in 2-dimensional space. And I found that, yes, you could discern some patterns. There were some things that explain the distribution of myxomycetes. I also, and the slide doesn't really show it, I started looking at niche, the concept of niche, and applying it to myxomycetes. And I think that was fairly successful. Another bump in the road occurred in 1987 when I received a Fulbright scholarship to go to India. I was actually surprised to get it because I was still fairly young in my career, fairly early, but I got it. I went to India and I tried to use that opportunity to maximum possible advantage. I spent the time at Himachal Pradesh University in Shimla, which is in northwestern India in the Himalayan foothills, where there are oak forests, and if you climb up higher elevations, there are spruce and fir forests. Well, that led ultimately, and you might recognize the name on that, one of the authors, to a biogeographical study of myxomycete distribution on basically continental scale. Indira spent a full ride in my lab. You saw a picture of her early in the week. She was a delightful person to work with. I've been very fortunate. I've hosted 7 Fulbright scholars. There might be one in the audience, but I don't know whether he remembers working in Fairmont or at that time, the University of Arkansas. I spent my career at 2 different universities, and the latter part of the last 20 years was the University of Arkansas. Before that, Fairmont State College, but it's now called a university. Then I got a grant from the National Geographic Society to do some field work in Alaska. Denali National Park is in Alaska, and if you ever go to Alaska, you certainly want to go there and take a look at it and all that. It's a neat place. Uh, Vegetation a little sparse in Alaska if you get a little up in elevation, but the forests tend to be dominated by two species of spruce. I won't go into which they are and whatever else, but there are not a lot of different trees. The vegetation is not incredibly diverse, which is actually a good comparative study looking at the vegetation or the ecosystems of other kinds, other places. That's a well-developed forest in Alaska. You can see the bryophyte cover on the ground. It's almost complete. A neat place. Some hazards. This is grizzly bear territory, and you don't really want to mess with grizzlies. And this particular study area of mine, which is south of Fairbanks, you could see grizzly bears leaving their marks on trees. So they were definitely there. So you always looked over your shoulder. This is north of the Brooks Range. I had an opportunity in 1989 to go north of the Brooks Range. You're looking south. This is July 20th, and it snowed overnight. So it'll give you an indication. Yeah, that's got to be cool. What you see in the foreground, I had nothing to do with it, is some instrumentation looking at measuring things, assessing that long ago global climate change. And the scientists who were working with that are already convinced, now that's a long time ago, well, a relatively long time ago, global climate change was occurring. My first introduction to the tropics, the neotropics, occurred in 1989, and I've worked out of this biological station, La Selva Biological Station in Costa Rica, a fair amount, although it's not the only place I work, but it's, if you want to get an introduction to the tropics, it's probably a good place to go. The infrastructure is excellent. That looks like a tropical forest. It is a tropical forest. It's different from that oak forest that you saw earlier because tropical forests are different from temperate forests. Are there myxos both places? Well, as all of you know, yes, there are. I was also intrigued with something that I pursued, a concept or a term that's applied here is canopy soil. We now know, and it's long been known, but it hasn't really been investigated for mycetozoans, myxomycetes and dictyostelids, that the amount of well, they're epiphytes, but they can be vascular epiphytes or bryophytes, that beneath those on branches and sometimes even on trunks of trees, there develops an organic layer, which I refer to as canopy soil. And in that soil, you can find myxomycetes and dictyostelids. And that soil, as it were, can be 40 meters above the ground. Kind of neat. This is a group in New Zealand where we were investigating. In fact, that's the first sort of expedition in which I was ever involved to go look at canopy soil. And the trees in New Zealand are pretty hard to, well, getting to the canopy. Many of the podocarps have a long trunk with the branches and leaves up top. It was a tough situation, but we were able to collect samples. Then, as a result of a conversation I had some years earlier with someone from the Australian Antarctic Division, I had an opportunity to go to the sub-Antarctic for the first time in 1995. This is an image from Macquarie Island. You'll see Macquarie Island on a map a little later, but you can see all those seals. Now, some of you are saying, what? No, they're not seals, they're penguins. Everyone recognizes penguins. Penguins can be incredibly abundant. The largest colony I saw in Macquarie had 250,000 penguins in one place. Pretty amazing. But sparse vegetation. There are only about 45 species of vascular plants on Macquarie Island. In any given community, you on this community, which is fairly rich for Macquarie, you'd have to go to the second hand if you're using your fingers to count. But for this community, the species richness is probably about 7. They're not very diverse. How do you get there? Well, you get there various ways, but you can't fly in, you can't walk in, you have to go by boat. And this is one of the vessels. I've actually been to the sub-Antarctic since then. This is one of the vessels that we went to the sub-Antarctic in. And he said, well, that's not very big. No, it is not. But that's all I could afford. The Southern Ocean can be incredibly rough. The first time I went down, we were operating in swells that were about 40 feet. Not in this vessel, somewhat larger vessel, but it's an incredible experience, to say the least. But you get there, I regard the subantarctic islands down in the Southern Ocean to be some of the more incredible places on Earth. They're raw, they're relatively free of human influence, and it's incredible. This is this is the main Auckland Island. It's a group of islands called the Auckland Islands. And on the East Coast, there are fjords, just like in Scandinavia. And this little fjord, we actually landed, and I did some collecting in the forest that you see there, but nice little waterfall spilling over. I mean, I like things like that. I've always been a field biologist. I always say, oh, that's more like it. That Hamia utricularis on the Auckland Islands, a subantarctic myxo, that's the largest fruiting I've ever seen for a myxomycete in the subantarctic. Fruitings do not tend to be very large. Well, I said you'd see a map. I've been working in the Southern Hemisphere quite a lot. And over the years, I've collected on the Auckland Islands. I've collected on the Campbell Islands. I've collected on Macquarie Island. In fact, I spent 4 months on Macquarie Island. I think I've pretty much got every myxomycete on the island, quite frankly. I mean, every day I could go in the field. I did go in the field over a period of 4 months. So I think I got most of them at least. Campbell Island, probably not. 2 trips to the Auckland Island, probably not. Stewart Island is just there are 3 islands constituting New Zealand, but you'll always hear of North Island, South Island, but there's a 3rd island, which is some size, called Stewart Island. So I've made a couple of trips there, but these are numbers of myxomycetes, or number of species known. And if you look at that, there's some evidence of a latitudinal gradient in terms of species richness in myxomycetes. Well, I went to New Zealand in 1992. I love the country, really love the country. And that does not look like West Virginia. It doesn't look like Arkansas. It doesn't look like Europe either. New Zealand is a very special place. It's not very large, but it has a reasonable diversity of vegetation. This shot was taken in the Southern Alps on the South Island. Ooh, this one too. You can see the Southern Alps in the background. He said, well, that looks like snow. Yes, there's snow. There's snow banks. And he said, well, if there's snow banks, you build a snowbank, they will come. You're right. What we call them. Here in the United States, we tend to use the term snowbank. I know you guys don't, but you build a snowbank and they will come. Snowbank myxomycetes. I also went to Australia in 1992, and the general conception of Australia is that it's very dry. Well, a lot of the continent is very dry. He said, well, that doesn't look like it's very favorable for myxomycetes. Well, it really isn't, but they're there. You can get them mostly through moist chamber culture work. There are field collections, but you may go for a whole day and never see something in the field. This is more typical Australia. That's a eucalyptus woodland. It looks quite dry. It is quite dry, but there are myxos there too. He said, whoa, that's pretty neat. This is in the Victorian Alps, south of Queensland. I would say they're beautiful. I really enjoyed collecting there. I'd go back in a moment, except I probably won't because of the expense and all involved, but a really neat place, and not what one visualizes as New Zealand. Well, making all those trips to New Zealand, I had an opportunity. And when you publish a book, and all of you have to be aware of this, you need a publisher, and a publisher needs a reason to publish it. They figure that they'll at least break even in terms of the profit they get, and they much prefer making money having the book published. And it's often very difficult to find a publisher. Well, I was lucky enough that I found a publisher for this book, and it was published. Not a heck of a lot of copies of it were, I think 200 copies were ever produced. And I would say it had modest at best success, but it was a book. I also actually, the people, I received a couple of grants from the government of Australia, and they even suggested, why not write a book on Australian myxomycetes? And I said, well, sure. That book took me a better part of 20 years to write. I'm not kidding you, 20 years to write. The publisher picked something to put on the cover that would not have been my first choice. He said, well, wait a minute, that's not a true myxomycete. Well, it still made the cover. Later in my career, well, actually the latter stages of my career, although beginning with Macquarie Island, I became more intrigued with the myxomycetes occurring on islands. In other words, isolated land masses in the middle of the ocean. This is Christmas Island. It's a really neat place to go, but it's in the Indian Ocean and not the easiest place to get to, but you get there and it's rather picturesque. You don't go there for the beaches. The coastline is pretty uniformly rugged. The forests are fairly intact, although they're mining phosphate on the island, so they're slowly denuding the island, but this part of the island is still intact. The forests are, well, different looking, and one of the big differences, they have rather large land crabs. I didn't include an image of one, but the land crabs ate the litter. They consumed the litter. So there are areas of the forest that are essentially bare ground in a lush tropical forest. It's really hard to find litter inhabiting myxomycetes because of that. This is another island, an island I went to very few years ago. This is Norfolk Island, which is where Norfolk Island pines come from. That seems very logical. A neat place, although I discovered that Norfolk Island pine decaying wood doesn't seem to be very good or very favorable for myxomycetes. They're there, but rather sparse. I also want to call attention to some people I've worked with over the years. I think I can identify all of them. Actually, I suspect that most of you could identify all of them, or most of them. Oh, goodness, that's Martin, whatever his name is. I don't remember the last name. Ura, I think most of you know. Oh, Carlos gave a presentation earlier in this Congress. The guy on the far left is one of my graduate students, Adam Rollins. He got his PhD working with myxomycetes, as did Carlos. Whoa, there's some more myxo people that some of you might recognize. Diana, my goodness, Arturo, and I believe that's Carlos, another Carlos. The guy in the middle is another one of my graduate students, Randy Dara. He's quite a photographer, and his photographs have graced many, well, a fair number of my publications. We're examining that weed in the foreground. So odd-looking little weed. Wait a minute, that's Welwitschia. If you're a botanist, you know what I'm talking about. It's Mecca. If you're a botanist, you've got to make a pilgrimage to see Welwitschia. Well, that's what I always thought. This is on the Macquarie Island. There were about 30 of us there for the 4 months. The guy in the red top, is Gary Larson, who was the other American with me on Macquarie Island. He's a full-fledged mycologist. In fact, we were supported on Macquarie Island to examine macrofungal biodiversity, but quite frankly, I spent more time trying to assess the myxomycetes, but these guys were willing to help me. I think you've seen some of those people before. In the Andes, about 15,000 feet. Yeah, there are myxos there. Wow. This is Argentina. You know, well, those folks really get around, don't they? Well, sometimes they do. The guy in the funny hat in the background is a mycologist, so we ignore him. The guy on the right on the foreground is Fred Spiegel, who works with protostelids. The guy on the left is Jim Cavender. If you've never met Jim Cavender, he's quite a character, and he's still the world's authority on dictyostelids. He's an incredible guy. The The guy behind him is one of his graduate students, but not something we consider for myxomycetes. Failure. We hate to admit failure. I think you agree with me on that. But sometimes it happens. In New Zealand, there are some volcanoes on the North Island that are snow-capped. In other words, you look in the background, we're on one of them. Those are nice snowbanks. My first trip to New Zealand to collect snowbank myxos, I didn't find anything on the North Island. Didn't look as much. Most of the, all the collections came from the South Island. But I said, my goodness, there got to be snowbank myxos on the North Island too. All I have to do is go at the right time of the year, and I'm going to fill my collecting box rather easily. Well, I did. Randy was with me as photographer and co-collector. We spent a couple of weeks. The timing was right. We knew that. We did not find a single snowbank myxomycetes, not a single one. He said, well, wow, that's real failure. It is. Do I regret it? No. Sometimes I've had collecting trips and I've come home with nothing. Now, this was a collecting trip to half a world away, and I came back with nothing, but just the experience, the sights, just being out there. Again, a field biologist loves that, eats it up. This is Clive Shirley, who's I hate to use the word amateur because in my experience, especially for macrofungi, I've met amateurs, if you want to use that term, and I think it's an inappropriate term, are far better, know more fungi than the so-called professionals. Let's say he's a non-professional myxomycetologist, and he's looking at the frond of a palm, looking for myxomycetes, and he's certainly the most active on the ground myxo person in New Zealand. There are not a lot of myxomycete people on the ground anywhere in the southern hemisphere, it seems. Wow, you've seen her before. This is in Fiordland, New Zealand. It's wet, rains, it's sopping wet. I have to commend her. She has been willing to follow me almost anywhere in the world. She did go to really bad places. She didn't go to Macquarie Island with me, but she's collected a lot of myxomycetes for me, willingly. And we're still married, and that's the thing that amazes me the most. Some advice. When you reach the twilight of your career, and I'm certainly there, I acknowledge that. I've reached the twilight of my career. Sometimes younger people will listen to you. Although that was more true decades ago than it is now. Younger people don't listen to older people anymore, right? You don't have to agree with me. I know it's true. One of the things, the most important things for us to do is pass on our knowledge and also our enthusiasm to the next generation. We have obligation to do it. This shot was taken in, 2006. And somebody said, well, that's Carlos Rojas, is it not? Yes. This is a workshop that we put on for junior high teachers and students, introducing them to myxomycetes, including, yes, if you look around, how to prepare moist chamber cultures. There was actually a pretty high level of enthusiasm. We did this a number of years. You got to get people started young, as it were. Now, they couldn't continue with it because I couldn't continue with it and Carlos couldn't continue with it. I doubt if their teachers continued with it, but at least made them aware of myxomycetes. They were ahead of me at the same age, remember? At that age in junior high, I would say, what are you talking about? Are you cursing me? Use the term myxomycete. This is a group of myxo enthusiasts, although they also include a couple people who are not. But if you look, he said, well, I recognize a few of those people. Wait a minute. I think David Mitchell is in there. Yep. And some other people you might recognize. For 5 years during the summer, I've taught a field course in Maine on myxomycetes. And it's actually drawn a reasonable number of people. That's what the course is about. It's a week-long course. You spent all day on myxos. Wow, that sounds bad. Well, maybe it sounds good to some people. And it's been a joy to do. I've thoroughly enjoyed it. And there are young people, and there are middle-aged people, and older people, and they all took the course. I've also had the opportunity over the years to host some people. Over a period of several years, I hosted 6 graduate students from India. Now, India is a place where not a lot of myxomycete work has been done recently, although there certainly is some history of doing decades ago. This is a young girl, a graduate student from India, in my lab, checking myxomycetes in the moist chamber culture. As I mentioned earlier, I've had 7 Fulbright scholars work in my lab, and several other people from work in my lab for months or even a year on other types of scholarships. And I like to think that they benefited from the experience. I did. I certainly benefited from working with them. This is a course that I taught in Thailand, the highest spot in Thailand where it rains almost all the time. This course was actually on macrofungi, but there's no way that I'm going to teach a course on macrofungi without introducing the myxomycetes and having them collect myxomycetes. It just goes with the territory. But a nice, enthusiastic group. This is earlier, actually in 2008. 2008. It's hard to remember all these dates. This is a group. We actually had a 3-day workshop in Thailand, in northern Thailand. And do some of those people benefit and go on? Well, yes. The third person from the left ultimately earned her PhD working with myxomycetes. So far as I know, the first person working with myxomycetes who got a PhD in Thailand. If you look at the right, the third person on the right, the guy, Thomas Edison de la Cruz from the Philippines, he ultimately got a PhD and actually visited me on a Fulbright. So they benefited. The young lady on the first row on the far right, Han Tran, got a degree working with myxomycetes. So you look at that and say, well, maybe there's a little bit of a legacy from that workshop. Ever give a workshop to a group of people who did not know very much English? Yes, I have. It's tough. The lady on the far left is Han Tran. This is Vietnam. Some of you might have figured that out. She translated for me, and I managed to get through the workshop. And again, a high level of enthusiasm. No, the people didn't know anything about myxos before the fact, but they learned something about it. This was another place that needs a lot of workshops. This is Africa. This shot was given at the university, or this workshop was given at the University of Nairobi. And yes, a level of enthusiasm, but no way to follow up. There's a lot of work that needs to be done on myxomycetes in Africa. There are a few little spots, but most of the continent, no. Now, admittedly, part of the reason not a lot of work has been done is the same reason that if you offered me an opportunity to go to the Middle East to collect myxomycetes, I'd turn you down. Some of Africa is pretty darn dangerous. I wouldn't go there. I wouldn't recommend anyone go there, but there's still a lot of myxos there that need to be documented, work that needs to be done. So again, I just reinforce that statement I made earlier. Educate, disseminate, disperse information, let other people know what you're doing. And the books that I've written some fairly technical books, but for the most part, I try to write a book that's understandable enough so that an educated layperson could pick it up and learn something about myxomycetes, or as the case may be, macrofungi. For a long time, well, in 1994, I published the book. Well, the publishing company, Timber Press, published the book, but I wrote it, called Myxomycetes, A Handbook of Slime Molds. Surprisingly, that book stayed in print until 2021, when the publisher finally decided to discontinue it. Yes, I used that little book in my workshops, and I think a lot of other people throughout the world have used it. I think it was a reasonably good book, but it's gone. Well, its brother or sister or whatever else is coming. In 2024, if nothing goes well, an expanded, larger, more pages, more pictures version of Myxomycetes, a Handbook of Slime Molds, is going to be published. You look at that and you say, wow, I'll rush out and get that. Well, I hope you do, because the publisher will enjoy that. But that's a mock cover. If you look at, search the bookshelves for that, you're not going to find it. It hasn't reached that stage. And what the Lone Pine Press did was develop a mock cover. I have no idea where they got that image of Myxomycetes, and not one I provide. They also spelled my name wrong. Wow. But I just wanted to alert you to the fact that, yes, there is another book coming, and I would certainly encourage you to at least take a look at it. Well, not thanks to you yet. When you do research on Myxomycetes, you make these trips to exotic places and do all these things, somebody's going to have to supply some funding. Well, 2 places have provided funding more often than any other places. NSF, the National Science Foundation of the United States, has really helped me a lot. But the National Geographic Society has helped me too. And those are the 2 places that I owe the most thanks. But I owe a lot of thanks to a lot of other people and organizations and whatever else. And wow, isn't that a pretty rainbow? Wow, somewhere over the rainbow, there are Myxomycetes. You got to keep that in mind. This is the general statement. If you travel a road for a long time, you're going to make a lot of stops and need the assistance of a lot of people to do what you want to accomplish. I cannot enough express my thanks to the many people who have helped me in my journey along this long and winding road. And it does include some people in the audience, and I especially thank you. Now, a totally different subject. I was given the instruction that or asked if I could include one more slide after my presentation. It's a letter from a former graduate student, Brandy Kluth, who is willing, or her university is willing, to host the next Congress in 2026. Other people are going to advocate this. I'm just going to give you some basic information. Brandy did not work with Myxomycetes. She worked with ecology and higher fungi. She knows quite a bit about Myxomycetes. She was in my lab. You couldn't walk into my lab without learning something about Myxomycetes. St. Peter's University is right at the edge of New York City, which gives it a real good advantage in terms of location, near 2 international airports. She's a very hardworking person, and if she indicates she can do something or will do something, she will. And I guess that's the highest recommendation anyone could ever give someone. So with that, I'm finished. You can go on and discuss the Congress or anything else. But thanks for giving me the opportunity.
Thank you, Stephen, for what?
Yeah.
Yeah, a little cameo. Thank you, Stephen, for the really impressive talk, and also for the introduction of the next venue of the Congress.
They've seen 3 slides of it. My wife just came in the room, and I was telling her, well, come on, let them see who you are.
Hello.
That's my field assistant.
Cheers. Yeah, we would like very much to see you here in person and to meet you, but still in Zoom. We're happy that you could join us that way. And let's hope maybe the next time we can meet in person. Thank you for the talk.
Okay.
So we are almost there. We are almost wrapping up our Congress. And but still we have yet another important task, and namely to choose the place for the next Congress. And by now we have one application, can I name it, or suggestion? But maybe somebody else has wants to advertise their institution, or is willing to propose some other venue. If there is somebody who wants to do that, so please, you're welcome to do that. But I see no hands raised. Is that so? Sorry, yeah. Well, it seems that we have the only proposal for the next Congress, right? Yeah. If nobody is, are there any comments in Zoom or anything? No, nothing. So that's then up to us to accept the proposal of Stephen's colleagues, right? What do you think of it? Oh, let's discuss that. So there is one hand, one vote for. That's not far from your place, right? All right. What do you think? I think we can just discuss it freely and then vote pro or contra. Is like a wedding? Is there anybody who has something opposed to this marriage? Well, if nobody, then raise your hands if you vote for it. Well, then decision is made, and then we're happy to announce that the next Congress will be held in the US, near New York. So yeah, and congratulations.